I'm Julia Souza, and I will be talking about gene mutations. A gene mutation is an alteration in a gene sequence. These can be as small as one gene block or a large segment of chromosomes. They're either inherited or they can be acquired throughout your lifespan. A mutation can occur 3 different ways: by substitution, deletions, or insertions. A substitution is when a nitrogenous base replaces the other. So that's when adenine, that's supposed to go with thymine, but in a mutation, it would go with either guanine or cytosine. A deletion is when one or more base pairs are lost in the DNA sequence. So, for example, if the order was supposed to be T, G, G, C, A, G, if the C and A were lost while the DNA was recombining or being replicated, that would be a deletion. An insertion is when one or more base pairs are added into the sequence. So, if the original sequence was supposed to be T, G, G, C, A, G, and then the mutated sequence would be T, G, G, T, A, T, C, A, G. And the TAT wasn't there originally, but it was added into the sequence. And mutations are caused by errors in the DNA replication or recombination, chemical damage to DNA, or radiation. What happens with a mutation is the genes are coded a certain way so that they can make proteins for your body, which is what's going to release the phenotype. But when a mutation occurs, a different protein will be made, which could either be good or bad. Some mutations are not even noticed because whatever protein they make is close enough to what it was originally supposed to make, but some of them have beneficial effects and some of them have harmful effects. Some of the beneficial mutations is that it will make a new version of the protein, which would help your body. It can increase chances of survival and reproducing in an organism, and it could also protect organisms from developing diseases. But on the other hand, harmful mutations can cause genetic disorders and cancer. Originally, mutations were thought to be permanent, but recently there has been a discovery that could change mutations, and that is CRISPR-Cas9. That's a form of gene editing. Basically, what this combination does is the Cas9 will cut out the unwanted mutated strand of DNA, and it will insert the CRISPR, which is the targeted strand. And that will fix the mutation, fixing a genetic disorder that is caused by it. And this has not yet been used on humans, but once it is, it can prevent many deaths due to genetic disorders.